Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
All right, today we are jumping right into applied pharmacogenomics. This is a field that's really changing the game for how we prescribe medications. We're finally starting to move away from that old one-size-fits-all approach and into an era of truly personalized medicine. And we're going to show you exactly how that works.
Look, this is a question every single one of us has asked in the clinic. You give two patients the same standard dose of a drug, and one gets better while the other ends up with serious side effects. Why? Well, more often than not, the answer is literally written in our patient's DNA. Pharmacogenomics is the key to unlocking that code. It lets us predict how someone will respond to a drug, helps us avoid those nasty adverse events, and ultimately lets us pick the right therapy from the get-go. So, here's our game plan. First, we'll start with the molecular basics, decoding the genetic blueprint. Then, we'll translate that genetic data into real clinical action. After that, we'll look at why genetic testing is now a must-have for some advanced therapies. And finally, we'll walk through the entire process a test has to survive to actually make it into your clinic. All right, first up, decoding the genetic blueprint. Before we can apply any of this in practice, we've got to go back to basics and understand the fundamental molecular biology that's driving everything. You know, it really all boils down to the central dogma of biology. It's a straight line from cause to effect. A small variation, a typo if you will, in a patient's DNA gets transcribed into RNA, which then gets translated into a protein. Now, if that protein happens to be a crucial drug metabolizing enzyme, that one little typo can mean everything. It can create an enzyme that works way too fast, clearing a drug before it has a chance to work, leading to treatment failure. Or it can create an enzyme that's too slow, causing the drug to build up to toxic levels. It's a direct, causal chain from a single gene to a real-world clinical outcome. The most common type of genetic typo we deal with is called a single nucleotide polymorphism, or SNP. Now, a missense SNP swaps out a single amino acid in the protein chain, and that can have a huge effect on how the protein folds and functions. But a nonsense SNP? Whoa, that's even more dramatic. It creates a premature stop sign, telling the cell to stop building the protein halfway through. The result is usually a completely non-functional protein.
Now, let's look at some even bigger changes. You have insertions or deletions, indels, that can cause a frame shift, which basically scrambles the entire protein sequence from that point on. Total loss of function. Then you have copy number variations, or CNVs, where entire genes get deleted or duplicated. A deletion, obviously, that's a total loss of function, but a duplication is really fascinating. This is the main reason we see the ultra-rapid metabolizer phenotype. These patients have extra copies of an enzyme gene, giving them supercharged drug metabolizing power.
So how on earth do we keep all this complexity straight in a busy clinic? Well, that's where the star allele system comes in. It's an absolute lifesaver. It's a brilliant shorthand that takes a whole block of variants that are inherited together, a haplotype, and bundles them into one simple functional name, like CYP2C93. A patient's diplotype is just their two inherited star alleles put together, and that simple combination lets us predict their clinical phenotype. It's really become the universal language of pharmacogenomics. Okay, to really make sense of a variant when you find one, you need the right tools. So think of this next bit as a quick and dirty bioinformatics tutorial. There are 3 public databases that are absolutely essential. DBSNP, which tells you population frequency, UniProt for protein function, and PDB for the 3D structure. Let's walk through a real case study with the important CYP2C9 3+ allele to see how this actually works. Step 1, we head over to DBSNP. And right away, you see something fascinating. The frequency of this variant is all over the map depending on the population. It's over 11% in South Asians, but barely 1% in people of African ancestry.
Now, this isn't just demographic trivia, right? This directly impacts our pretest probability and helps us figure out how cost-effective a screening program would be for different patient populations.
Now for step 2. UniPro acts as our bridge. It takes that raw DNA variant and translates it into the specific protein level consequence. In this case, an isoleucine is swapped for a leucine at position 359. But here's the best part. It gives you expertly curated notes confirming that this variant is linked to impaired metabolism and the poor metabolizer phenotype for critical drugs like warfarin.
And finally, PDB gives us the why. You can literally see it. We get a 3D view of the protein. And this is so cool. The mutation is nowhere near the active site where the drug binds. But what it does is cause these long-range ripple effect changes in the protein's shape that mess up the channel the drug uses to get in. It's just a fantastic example of how a protein is a dynamic allosteric machine. Okay, so we've cracked the genetic code. Now what do we do with it? That brings us to our second section, from genotype to phenotype. This is where the rubber meets the road, translating that raw data into a prediction that actually guides our decisions in the clinic. The way we translate genotype to phenotype is actually pretty elegant. We use an activity score system. Each allele gets a score. A normal function allele gets a 1, a reduced function 1 gets a half point, 0.5, and a no function allele gets a big fat 0. We just add the scores from the two alleles together to get a final diplotype score. That score then neatly sorts the patient into one of 4 categories: poor, intermediate, normal, or ultra-rapid metabolizer. Simple as that. Now, this is a concept you absolutely have to understand because the clinical consequence is a complete flip-flop depending on the drug. Let's take a poor metabolizer. If you give them a standard active drug, they can't clear it, and they're at high risk for toxicity. But if you give them a prodrug like codeine, which needs that enzyme to be activated, they get 0 pain relief. For an ultra-rapid metabolizer, it's the exact opposite. They might clear an active drug so fast it doesn't work, but that same dose of codeine could lead to a toxic overdose. It's a critical pharmacology dichotomy. And listen, it's not all about metabolic enzymes. Drug transporters are just as important. A perfect example is the SLCO1B1 transporter, which is responsible for pulling statins into the liver. If a patient has a variant that reduces the transporter's function, less statin gets into the liver and more of it stays in the bloodstream. This dramatically jacks up the risk for statin-induced myopathy. The end result for the patient looks exactly the same as if they had a metabolic defect. Okay, let's shift gears. In our third section, we're moving into a realm where genetic testing isn't just helpful or a good idea, it's mandatory. It's an absolute prerequisite before you can even think about writing the prescription. For these advanced biologic drugs, you can think of mandatory testing as resting on 3 strategic pillars. First, there's safety screening. We have to test for the HLA-B5701 allele before giving the HIV drug abacavir to prevent a potentially deadly hypersensitivity reaction. Second, target identification. We test for HER2 amplification in breast cancer to make sure the patient will actually respond to trastuzumab. And third, resistance assessment. We test for KRAS mutations in colorectal cancer to avoid giving a useless and expensive anti-EGFR therapy to patients who won't benefit. This principle also extends to the cutting edge of medicine, cell and gene therapies, but with a really important twist in the rationale. For a gene replacement therapy like Zolgensma, the genetic test is diagnostic. We're confirming the patient has the inherited genetic defect that the therapy is designed to fix. But for CAR T therapy, the test is molecular pathology. We're not looking at the patient's inherited DNA. We're testing the tumor itself to confirm it has the protein target, like CD19 on its surface. 2 totally different questions. So we have all of this incredible science. But how does a test actually get from a research paper into your day-to-day practice? Well, that brings us to our final section, the path to the clinic, and the journey through what's often called the translational gauntlet. For any genetic test to make it through that gauntlet and be adopted, it has to pass a super rigorous evidence-based evaluation called the ACCE framework. Think of it as a 4-part exam. First, analytical validity. Does the lab test actually work reliably? Second, clinical validity. Does the genetic variant consistently and accurately predict the clinical outcome? Third, and this is the big one, clinical utility. Does using this test in the real world actually improve patient outcomes? And finally, ELSI. Have we seriously thought through all the ethical, legal, and social implications? And that last part, ELSE, is absolutely not just some box-ticking exercise at the end. These are massive hurdles that can stop a test in its tracks.
we have to proactively deal with major barriers like the potential for genetic discrimination, which laws like GINA in the US are designed to prevent. We need rock-solid systems for privacy and confidentiality, and the informed consent process has to be incredibly clear and comprehensive. So the journey from a single line of genetic code to a real clinical decision is a long and challenging one. But it's a path that is defining the future of precision medicine right now. The biggest challenge is no longer just our ability to generate the data, but our ability to translate it into evidence-based, effective, and ethical patient care. So I'll leave you with this question. Now that you've seen the whole path from code to clinic, what's the first step you're going to take to integrate pharmacogenomics into your own practice?
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.